Hello everyone, welcome to our new channel KNK Knowledge Hub. And now we are going to discuss about the simple staining procedure, a very important procedure in microbiology practicals. So let's see what is that. So first here if we are going to listen this simple staining procedure means what what are the elements we can able to get means we can able to know how can we perform the staining and what is the principle behind and what are the requirements we need and what is the procedure behind and how can we interpret the results after the procedure is done. Then, see the first one, what is simple staining? It is a technique that is used to observe the morphological characteristics of bacterial cells. By this technique, we can able to study the shape, arrangement of bacterial cells. It is a basic staining method in microbiology. Then coming to the principle behind simple staining. In simple staining, bacterial smear is stained with a single reagent which produces a distinctive contrast between the organism and its bright background. Basic stains such as methylene blue, crystal violet with a positively charged chromogen are preferred because bacterial nucleic acids and certain cell wall components carry a negative charge which will strongly attracts and binds to the cationic chromogen. And the purpose of simple staining is to elucidate the morphology and arrangement of bacterial cells. So the most commonly used basic stains are methylene blue, crystal violet and saffronin. Then coming to the requirements. We need clean glass slide and we need any type of basic stain like we can go with crystal violet or methylene blue or saffronin, and we need a microscope, we need inoculation loop, we need bacterial culture and we need some distilled water. Then coming to the procedure behind, a clean glass slide is taken and bacterial inoculum is placed on it, and with the help of inoculation loop, a thin smear is made. Then, bacterial smear is allowed to air dry. After air dry, bacterial smear is flooded with basic stain such as methylene blue or crystal violet or saffronin. Allow the smear for 1 to 2 minutes to uptake the stain. After, place the slide under running tap water and gently wash the excess of stain. Let the slide air dry and observe under the microscope. So when you observe under the microscope, bacterial cells can be observed colorfully under the microscope and the background will be in bright. And their morphology in the sense what type of shape they are having, whether they are in rod shaped or spherical shaped. And the arrangement of rods, some rods are in chains and some rods are in palisade arrangement or cocci. If they are spherical in shape, we call them as cocci, and if the cocci are in chains or if the cocci are in clusters, we need to observe. Then according to the observation, the bacterial morphology can be noted. Here I have given some examples to see what is the arrangement. Like if you observe the arrangement of the cells, like rod shaped cells which are arranged in chains, you can say streptobacilli. Cocci, that means spherical shaped cells, if they are in clusters, you can say they are staphylococci. Cocci in chains, you can say those are streptococci. You can see here some pictures. So this is the cluster arrangement of cocci. So you call it as a staphylococci. And this is chain-like arrangement of cocci. So you can say that is streptococci. And here these cells are rod-shaped cells which are arranged in a chain-like manner. So you can call it as streptobacilli. So like this, we can able to interpret the results after your observation. Thank you.